Hello everyone and welcome to the Chief Science Officer's Zoom in on Science Calls, where we learn about STEM professionals and their careers to understand the impact of their job in modern life. My name is Yusuf Fatima from Kenya and I'm personally excited to host today's call. So for all of us to enjoy this experience, please mute your microphones, post your questions at any time during the presentation in the chat box. We'll be sure to have time after the slides to answer as many as possible. Now on past calls, we have learned a lot different things, and today we celebrate protein synthesis. So now please welcome Sydney to Zoom in on Science, focusing on biochemistry. Sid, can you tell our participants a little bit about yourself and your work? Please share your slides, and remember participants, post your questions in the chat.
Welcome. I'm delighted to have the chance to talk with you all. I'm speaking to you from Arizona State University, where I'm employed as a scientist. I'm a professor. I'm the director of a center in what we call the Biodesign Institute. And my center is the Center for Bioenergetics, and we work on a few different topics. I'm a professor in what people used to call chemistry. Now we call it the School of Molecular Sciences, but it's the chemistry department. And we have a number of topics, as I mentioned, that we work on. And I'm going to bring up some slides to show you what we work on and to talk a bit about the basics of one of the areas, which is protein synthesis. Topics that we work on are essentially at the moment are threefold. One of the topics is involved in the detection of something that's very timely at the moment, which is in the detection of viral RNAs. And we've developed a nanoparticle, a nucleic acid, a DNA nanoparticle, that actually is able to intercept and bind with the conserved regions of RNAs from all sorts of retroviruses. And the one that we're mainly working on at the moment, in which we can detect all of the serotypes, is dengue virus. But we can look at pretty much anything, and certainly we're easing into the business of looking at coronaviruses, as probably is not too great a surprise to you. So that's one of the topics, but it's not something that I'm really going to focus on today. A second, which has been a bigger effort on our part, is disease-oriented. And it is intended to allow us to treat a type of disease, a disease of the mitochondria. The mitochondria, as some of you probably know, are the energy-producing organelles, the energy-producing part of all mammalian, all eukaryotic cells, really, and produce much of the ATP that allows us to survive and to do things like moving our fingers and walking around, and literally provides the energy to allow us to do all of that. We've tried to use our understanding of the mechanism by which the ATP is produced to produce compounds that allow us to augment ATP production and to suppress some of the negative effects of mitochondrial dysfunction. There are hundreds of mitochondrial diseases, and we're focused on trying to create drugs that can ameliorate the effects of mitochondrial diseases. But what I'll talk about today is protein synthesis, which has been our longest existing program, in which we make proteins, arrays of connected amino acids, sometimes hundreds or thousands of them, that contain within them one or two amino acids that are not natural, that are artificial, that we've learned to insert into the protein to study the mechanism or to alter its effects. And I'm going to talk about some very simple part of that today, which is related to something that many of you have probably heard of, the central dogma of molecular biology, shown here in its simplest form, in which DNA, by replication, reproduces itself in a cell. By transcription, makes RNA that contains the information from the DNA, and where the RNA participates in translation to make functional molecules, molecules that carry out metabolic reactions, that are structural molecules, that support huge numbers of biological functions. And I'm going to talk just a little bit about the synthesis of proteins, the mechanism which they're synthesized. And today, I'm only going to talk about the way that proteins are synthesized in simple organisms, typically in bacteria, and their synthesis from RNA. Now, in truth, they're actually made from 3 different kinds of RNA. They're made from transfer RNAs. There's at least one transfer RNA for every amino acid that nature uses in proteins. For those of you who have some molecular orientation, this is the structure of a single amino acid attached to its cognate, to its matching transfer RNA. That's one of the RNAs that's used in protein synthesis. The second is a mixture of RNAs and proteins called the ribosome. It has a large subunit, which contains 2 RNAs, and a small subunit, which contains 1 RNA. The large subunit also contains about 30 proteins, and the small subunit about 20 proteins. And this is the assembly of the 2 subunits of the ribosome with a site that will allow a messenger RNA, the 3rd kind of RNA, to interact and to result in protein synthesis. Let me point out that the assembled ribosome has 3 sites. There's an A site shown here for the aminoacyl tRNA, that's this species. There's a P site for a matching tRNA that has another amino acid attached to it. And there's an E site, which is part of the overall mechanism. So 3 RNAs work in concert to make protein. This is a slide to illustrate how we start protein synthesis. We take one of the 2 subunits, the smaller of the 2 subunits, and we associate protein factors with it. And additionally, a molecule that, like ATP, is an energy molecule, it's GTP, and it forms an initial complex which binds the tRNA that you saw on the previous slide, having a single amino acid on it. That single amino acid is brought to a specific site in the messenger. And initiation of protein synthesis requires that the correct site be used. And so this initial aggregate, what's called sometimes a pre-initiation complex, then binds to the other ribosomal subunit. As part of that binding process, you need to pay energy to bind them together, and the energy is paid for by hydrolyzing a GTP molecule, by losing one of its 3 phosphates to give GDP, and you wind up with the initiation complex, which is shown here. So the initiation complex is the full starting complex for making proteins. It has 3 types of RNA. One of the types of RNA is not shown here. It's within these green blobs, which are the 2 subunits of the ribosome. There's a second RNA, which is in the transfer RNA, which contains the first amino acid. And there's the 3rd kind of RNA, the messenger RNA, which has individual nucleotides, the sequence of which determine the tRNA that is introduced to the ribosome. So in a moment, we're going to introduce another tRNA into this A site. And those 2 tRNAs are going to react together to make the first bond that leads the amino acids to be zipped together into a protein. There's some narrative here that basically says, in a lot of words, what I just said. And the correct site is determined by 2 things. It's determined by the sequence of nucleotides that binds this tRNA it's always an AUG in bacteria, and it always binds a tRNA which has the amino acid methionine attached to it. The site that's the authentic start site was determined in part by a sequence upstream from this AUG that binds to the RNA that's within the messenger RNA. That site in the messenger RNA is shown up here. It's called the Shine-Delgarno sequence, and it aligns the correct AUG with the start site for protein synthesis. So through this process, which uses energy, it uses 3 initiation factors, it uses 2 ribosomal subunits, it uses a tRNA containing a single amino acid, we find our way through a pre-initiation complex all the way to the starting complex for protein synthesis. And I'm going to go on now to show you what happens from this starting complex in my final slide. And that's shown here. It's a little busy, but let's take a look. So this is where we were in the previous slide. And in the previous slide, we are missing anything in the empty A site. And so we introduce another aminoacyl tRNA into that A site, and it takes a protein factor to introduce it. That protein factor is called an elongation factor. It's elongation factor TU. Cells need a lot of this elongation factor because they make a lot of proteins, and every transfer RNA has to be brought to the A site by elongation factor TU. It comes with GTP, it's an energy source, so you have to pay energy to introduce the second tRNA into the A site in the presence of EFTU. Because so much protein needs to be made, EFTU is the most abundant protein in E. coli. It's 5% of all protein in E. coli. After you use the EFTU to introduce the tRNA, there's a recycling mechanism for EFTU, which I'm not going to dwell on. And we go on with 2 tRNAs, a methionine tRNA and a second tRNA, the first one in the P site, the second one in the A site. They react together to form a peptide bond on the A site, transferring this amino acid from the P site tRNA to the A site. We form a dipeptide, the first peptide bond in the protein. We then go on to move this dipeptide relative to the ribosome. It stays in the same place on the messenger RNA, but the ribosome moves. And so now we have an empty E site, which has been opened up by another elongation factor, EFG, which also requires energy. And now we move the empty tRNA that used to have methionine into the E site. In the new P site, we have the tRNA on which we've made a dipeptide. And as EFG exits this complex, we cycle back to something that looked exactly like the starting material, except that instead of having 1 amino acid on this tRNA, we now have 2. And this tRNA with the 2 amino acids goes through the next peptide bond forming cycle to add a 3rd amino acid, and then a 4th, and then a 5th, and 30 or 50 or 322, however many are required. So how do we stop? Well, we stop because at some point in the message, there's a stop signal. And the stop signal is 3 nucleotides, just like these, that are either the letters UAG, UAA, or UGA. And in a normal cell, there is no tRNA that corresponds to that codon. It's an empty codon. And so when that codon appears, there's a special factor, a release factor. There's two of them in E. coli that are important. And those two release factors result in removal of the protein from the ribosome. And through this repetitive cycle, we can make a protein of any length that we need to make. So that's the story that I have for you today, and I'd be happy to answer some questions if you have them.
Thank you very much for telling us about yourself. It's evident that you're truly passionate about your work. So while we're learning about what you do, participants have left questions in the chat, but let's start with a couple of questions I had prepared. Now, I believe you have already answered the 3 questions I had prepared, which were just on the topic of the mechanism of how protein synthesis starts and ends, and why there's so much elongation factor TU in bacterial cells. So I'm just going to go to another one, which I had prepared, and it's who was your mentor as a child?
Who was my mentor as a child? That's not a chemistry question. I guess I would say that my mentor as a child were my parents.
Like parents really do play an important role in like shaping the adult version of you. So another question I had prepared, what advice would you give to your 14-year-old self?
The advice that I would give, you can do many things in life, and you need to decide what's important to you. Because to do something significant takes a lot of effort. And you should only do something, if you have the choice, you should only do something that you want to do, something that you like to do, something that you can work hard on and do well. Because if you're going to spend a lot of your life doing it, you should try and do it the best you can.
Wow, that's very inspirational. So now let's move on to the participant questions. So Cecil Austin from Pennsylvania asks, have you ever experimented with creating proteins that contain 3 amino acids that do not exist naturally? Does this cause a new function, or do all combinations of proteins already exist naturally?
There are many amino acids, most amino acids, that do not exist naturally. My laboratory has looked at a number of proteins that have 2 unnatural amino acids so that we can watch them interact or reinforce each other's action. My lab has actually never looked at a protein into which we have purposefully added multiple 3, 4, 5 amino acids that are not naturally occurring. That said, there are laboratories that have done that. There are laboratories that have made relatively short polypeptides, maybe they're proteins, which have several reassigned amino acids, a number of which do not exist in nature. And today, mostly they have done that to show that they can do it. So that if they find a reason for wanting to introduce 3 or 5 amino acids into a protein, they'll know how to do it. And you ask a good question, because someday, probably not all that far from now, we're going to have the information, the techniques, the knowledge for the design of proteins that will make us very likely want to add multiple non-natural proteins. Does that help?
Yeah. Okay, so Kelly from CSO International asks, how long have you been a professor at ASU?
I've been a professor at ASU since 2008. Before that, I was a professor at the University of Virginia for off and on for 30 years. And before that, believe it or not, I was a professor at Massachusetts Institute of Technology for 8 or 9 years. So I've been a professor for a long time.
Yes, yes, you have. Okay, another question Kelly asked is, what has been your favorite topics to research?
The favorite topics to, I didn't catch the last word.
Research, favorite topics to research.
I see, okay. So the topics that I've researched the longest actually is protein synthesis. and which I still work on, and we still have a number of people who do chemistry and biochemistry in this system. We still publish, 2 or 5 papers a year in this area. There was another subject that I worked on for a long time, but have now sort of stopped for complicated reasons. And it was on an anti-tumor agent called bleomycin, which is used in the clinic to treat a variety of squamous cell carcinomas and malignant lymphomas. We did the first total synthesis of bleomycin. We did a solid phase synthesis of bleomycin. We've made combinatorial libraries of bleomycins, and we've contributed importantly to the mechanism of action of that drug. So we're very happy with what we did. We could probably still find things to do, but there are other challenges, and we've taken those up instead.
Okay, so moving on to the next question. Siyoso Tyra from Central Region asks, what piece of advice or quotes have you used to complete your work to its fullest potential?
What quote? If there's anything any quote that always comes to mind as regards professional things, as regards research and other professional things, if there's something that's worth doing, it's worth doing well. And that isn't something that I made up, and I don't know who first said it, but I think that's those are words to follow. If you're going to spend your time doing something, doing it as best you can is the thing to do.
100% agreed. Siyoso Tyra also asks, did you know you always wanted to end up here when going through school, or did you move around?
No, I changed fields. I started college. I was, and I hope I still am, quite good at mathematics. And I went to school to become a math major. And I was a math major for a while. And then as I traveled along, just as I told you before this meeting started, that chemistry changes as you go along. Well, math changes as well. And math changed from things that I thought pretty good at to things that I was okay at. And I thought I could choose something that I would learn to be better at. And so I moved purposefully from math to chemistry while I was a sophomore in college.
Wow. Okay, so Kelly from CSO International asks, how many students on average do you have in your lab at Biodesign Institute each semester?
So the number of coworkers that I have varies as a function of funding, as is true for virtually every laboratory in the United States. At the moment, depending on how you count, if you count everybody, we have fewer than 10 people in the laboratory. There have been years when I've had 35 people in the laboratory, but those are in years when those who manage funding are more bountiful to all of us than they have been in the last 5 or 10 years.
Wow. See, a lot of people are probably interested with biochemistry, so, well, I am. Okay, another question is from CSO Austin, and he asks, are there any mysteries that exist revolving around cell replication / protein synthesis?
Yeah, there are endless numbers of them. And not only are there endless numbers of them, but for many of them, we don't even know what they are yet. So I'll give you an example. I mean, the ribosome has been studied intensively, and it's been studied because one of the things that differentiates bacteria from people is their ribosomes. The ribosomal architectures are dramatically different, and so there are small molecules that bind to bacterial ribosomes that don't bind to human ribosomes. And you can use the targeting of those ribosomes to kill the bacteria without harming the host. So one issue is, what are the molecules in future generations of molecules that will be able to selectively manage interaction with bacterial ribosomes as compared with human ribosomes. It's not clear. Bacteria, although they're small and at one level fragile, vastly outnumber us. And they're very good at mutating. When you have a generation time of 20 minutes, you can change your basic nature and the small molecules that you're susceptible to in a very short period of time. And bacteria tend to do that. And so we constantly need new antibacterial agents. And it's hard to find them. And it's really hard to find them. And it's a mystery what the best way is to go about finding new ones. At a more fundamental level, there are many things about the mechanism of protein biosynthesis, the rate limiting steps, the selectivities, the maximum size of an amino acid that can be incorporated into a ribosome, how you could change a ribosome, and nature does change its ribosomes, how you could change ribosomes to make things that are different kinds of polypeptides, different kinds of proteins. None of those things are understood. And what's limiting in our lack of understanding, even that isn't understood. So for all we know about protein synthesis, we mostly don't know about protein synthesis.
Doesn't that get confusing at times? Like the last bit you said, what we know is what we don't know.
Well, you can ask questions. And my laboratory, for example, I mean, there are structural similarities between all the amino acids that nature uses. And for years, because we wanted to put in amino acids that didn't occur in nature, it was a source of frustration to us. And then at one point, I simply said, I think we need to take the ribosome and we need to re-engineer it. And we need to find a method to isolate modified ribosomes that still work, but accept a broader spectrum of amino acids. And actually, that's what we do. And we've learned a lot about how the ribosome does business by having done that. So you can turn it around and you can use it as a device for learning things at the expense of your time, of course. But you can learn new things about the ribosome and cause the ribosome to do new things that are potentially interesting and valuable.
Wow. Okay, so CSO Chipani from Kenya asks, what's your favorite thing to have done, or job or project?
Oh, I've done a number of things that required a large effort by a lot of people. And I was only distinguished in the process by being responsible for the whole thing. But we synthesized bleomycin, as I've told you, which at the time we made it was the biggest non-regular molecule that anybody had ever made. And I felt good about that. We've learned to put into proteins extraordinarily different kinds of amino acids that create new functions in proteins. And we've learned to do it not just in a test tube, but inside a bacterium. And we think it will be easily possible to do it inside a mammalian cell. And I'm excited about that. So as time goes on, you do different things and you get excited about them. I helped discover a new clinically used anti-tumor agent. I felt good about that.
Wow, that's actually really exciting. I think you've gotten us all really excited to see where that journey takes you. So CSO Tyre from Central Region asks, how has your job adapted during this pandemic? And do you prefer this lifestyle or your previous one?
Oh, this is, it's a lifestyle in which I can survive and I can get things done, but I wouldn't describe it as a good time. You get used to roaming around. I spent literally 50 years of my life traveling more or less continuously, and I haven't gone anywhere in several months, which is already a record for me. And by the time it's done, I think it's going to be a much bigger record. No, this is not what I would describe as an ideal alternative lifestyle. But it is one that allows me to stay productive. It allows me to keep moving. It gets through the day, and it's not generally terribly unpleasant, but it is very confining.
Yeah, 100%. We've been stuck at home for like 2 months now. Haven't
Right.
gone to school.
Right. I'm
But
in day 70 something.
I think we all are, but hopefully all goes well.
Yep. That is my hope.
So I think that's all for the participant questions. But before we close this call, I just have one final question. What advice would you give to us as young leaders for our future?
The answer would be just a slightly perhaps different version of the one that I gave you earlier. There are many things that you can do with your life. There are many choices in front of you. And if you want to accomplish something, it's going to require a lot of your time. And because it's going to require a lot of your time, you should choose something that you really want to do. And be as certain as you can about the fact that it's really what you want to do, and then do the best you can at it.
Yeah, I'm definitely taking that to heart. That's very inspiring. Thank you. Okay, so thank you so much for joining us today, Sid. You're an inspiration to many of us, and we hope you can participate in another call someday as you continue to zoom in on science. Now, CSOs, please unmute your microphones and we will close with our motto. Okay, so CSOs, don't just hope it happens.
Make it happen.
Thank you all so much for joining. I hope you have an amazing day ahead of you, or good night's sleep wherever you are in the world.
Thank you so much. I've had a very good visit with you, and I appreciate it.
And yeah, this has been so much fun, and we've learned so much from you, Sid. Thank you again.
We know we can learn so much more. Austin probably has 15 more questions. We can't wait to hear how you all do on your exam on protein synthesis. So let me know and I'll let Sid know. But Sid, we look forward to visiting ASU and your laboratory in the future as well.
Great.
Thank you so much.
Sooner rather than later.
That's right. Thank you all for joining the call in Kenya. Awesome. You guys need to go to bed. All right, until next time.
Bye. Bye now.